Welcome back, everybody. Today we are going to discuss biomolecules in this summary series. This chapter is of great importance from your board exam perspective. This chapter involves a study of entire molecules that are of great importance involved in human body. As the name suggests, biomolecules. We will be discussing all those molecules that occur in our body. Also, how they occur, how we obtain that in the laboratory, how we synthesize them, how we elucidate their structures. So you can expect at least 1 to 2 questions in your board examination. So let us begin, everybody. What are we going to discuss? We will be discussing many polysaccharides and monosaccharides in starting from glucose, sucrose, maltose and lactose. Then we'll be discussing amino acids. We also will be discussing nucleic acids, protein and nitrogenous bases. So I welcome you all guys in this summary series. My name is Dr. Lakshay Kathuria. Let us start with very first topic for today, that is glucose. What is a glucose? You know, glucose is the source of energy to us, and it is a carbohydrate. Carbohydrates are generally sweet in taste. What are they, guys? They are sweet in taste, and they play an immense role in biochemistry. Biochemistry is a branch of science, branch of chemistry, which deals with the molecules present in human body and any animal body for the sake of discussion. Glucose is one of the most abundant substance among all the biomolecules. So this becomes important to study what is actually glucose, what is its molecular formula, how we obtain it, what is the source, all those things. So let's begin, everybody. What is a glucose and how do we actually prepare that? So you know sucrose very well, which is obtained from cane sugar. That is also known as cane sugar. So the other name for sucrose is cane sugar. Sometime in your board examinations or any sort of competitive examinations, if you see cane sugar, it means sucrose. What's the formula for sucrose, guys? It's C12H22, C12H22O11. When you take a solution of sucrose in water and add some drops of acid, for example dilute HCl, this will break down into its precursors. This is a disaccharide. Disaccharide means it is made up of two sugar molecules, two carbohydrate, two monosaccharide units. Those two saccharide units are glucose and fructose. So we can say that glucose and fructose together make up sucrose. And how we obtain that? The source for glucose and fructose is sucrose. So when you consume sucrose, your body has glucose and fructose after its hydrolysis, acidic hydrolysis. This hydrolysis is catalyzed by acid present. Okay, and how do we commercially obtain glucose? That is done by starch, starting from starch. So starch will be, or we can take cellulose. When you add little bit of acid to it at this temperature approximately of 2 to 3 atm pressure, we can obtain glucose units. So sucrose is just made up of glucose units. So let us try to understand what is its structure, what is its molecular formula, how we obtain it, what was done to figure out to elucidate its structure in the history also. So its molecular formula, as we have just seen, it's C6H12O6. If I ask you to write its empirical formula, do you see there is a 6 common factor in all the 3 atoms here? So we can write C with H2 and oxygen. This is the empirical formula for this molecule. And what will be the common factor here? 6 is the common factor. 6 into 2, hydrogen 12, oxygen R6. So by the help of mole concept, we know mole concept is one of the important chapter in 11th grade. It's not just one of the chapter, it is the language of chemistry. If you do not know what is mole concept, I recommend you people to also be ready with mole concept, because wherever you go in chemistry, mole concept will be after you. It will follow you. So using mole concept like percentage, mass percentage, or combustion method, combustion reaction. What is combustion reaction? When you burn any hydrocarbon in the presence of oxygen, every carbon atom will transform itself to CO2 and every hydrogen atom will be transformed to water molecules. So we can then figure out how much of carbon, how much of hydrogen is present in this formula. Okay, so This is the overall, this is the general formula for general skeleton of glucose molecule. You can see that it has an aldehyde functional group. What is this, guys? CHO represent aldehyde. You are very well aware of it. And there are 4 hydroxy present. There are 4 alcohol units present in this molecule. There is another alcohol present, so total 5. But out of 5, 4 of them are secondary alcohol and this one is primary alcohol. HI is hydroiodic acid. When you treat these two together and heat it, what happens? Entire oxygen containing carbon, so all of these oxygen, they all carbon which is present in very high oxidation state, they all get reduced, they all undergo reduction and you obtain n-hexane. Once we figured out that all the carbon atoms are connected in a straight chain, this gives us a hint, this gives us an evidence that all these carbon atoms in glucose must also be connected one after the another in a straight chain. So this becomes one of your question, how do we actually know that carbon atoms are connected in a straight chain one after the another? 6 carbon atoms are connected in glucose molecule. This is the experiment. So this becomes your question. Question would be like that, how do you know that 6 carbons are attached in a straight chain? When you react glucose with HI and heat it, prolonged heating, you have to heat it for many days. For many days you have to heat it, and that proves all the carbon atoms are connected in a straight chain. Question number 1. High chance that this would be asked in examination. After that, there would be another question, that how do we actually know that there is an aldehyde group present? Why not a ketone group? Why not any other kind of functional group present in the glucose framework? So whenever an aldehyde is reacted with bromine water, Br2H2O, bromine water, this CHO will be oxidized to its corresponding acid. Rest all these alcohol units are intact. They are untouched. Nothing happens to these alcohol unit. So if you take an aldose, what is the meaning of aldose? Any sugar molecule, if it has aldehyde unit present in it, that is given a name, in general, aldose. Treating an aldose with mild oxidizing agent like Br2 water, bromine water, will convert aldose to aldonic acid. What is the aldose is this here? This is glucose. So glucose will be converted to gluconic acid. So when scientists tried to figure out the structure of this glucose unit, they already were known to the fact that there is something called gluconic acid. So they compared it with gluconic acid and they found that actually there was a COOH produced, which could be only possible to obtain from CHO if at all it is present in glucose framework. So this becomes another question, how do you know there is aldehyde group present? How did you make sure? How did you make sure that there is an aldehyde unit present in glucose? This is one of the questions. So make sure you know this, and you should be able to write down this expression, this equation, and also the comments. After that, we need to figure out of many alcohol units, how many of them are primary, how many of them are secondary or tertiary kind of alcohol. So when you react it with concentrated HNO3, you should know concentrated HNO3 is a strong oxidizing agent that will oxidize primary alcohol to its corresponding acid. Check this please, the secondary alcohols are untouched. They are not changed. They are as it is. So primary alcohol is changed. What is the change in the oxidation state? How many oxygen atoms are now extra in this molecule? What has happened to glucose? It has got transformed to glucaric acid. So this makes us again sure of the presence of just one primary alcohol in this entire glucose framework. That's again another question for you guys. This confirms the presence of primary alcohol. How many primary alcohol units are present? Just one. There are another 4 alcoholic units present. How do we actually make sure that there are 4 more alcohol present? So what is ACO? Let me first explain AC kya hai. What is AC? AC represent acetyl. When you take acetic anhydride, and treat it with an alcohol, so what is going to happen eventually? Let me write a general example. When an alcohol attacks on this center, this whole group leaves. This is known as a leaving group. So the compound formed would be, an alcohol will be converted to R oxygen C double bond O CH3. This is the compound obtained. So I am going to name this entire unit as OAC or OAC. So the alcohol is converted to its acetate. The alcohol is converted to its acetate, OAC. How many alcohols are present here, guys? 5 alcohols are present. 5 alcoholic functional groups are present. And we were already sure about that there is one secondary alcohol present. Treating it with acetic anhydride, we obtain that all these alcoholic units are converted to their corresponding esters. Corresponding esters. Please check, this is an ester molecule. Esters are sweet smelling compounds. They are fragnating compounds. So all of these are obtained. So what's the product? Pentaester. Why pentaester? Because there are 5 OH groups present. Okay. This is the structure of AC2O. This is the structure of AC. If I connect oxygen over here, it becomes ACO. So in short, can you tell me in the comment box what will be the name of this molecule? Sir, this looks like AC. This looks like ACO. This looks like ACOH. Right? Okay, great. So anhydride plus an alcohol will result into ester and acid. This part will transform to an acid. CH3COOH will be an acid, acetic acid, which is in general in commercial naming known as acetic acid or vinegar, for example. Okay, guys. Very good. So that is another question. How do we identify other alcoholic units present in glucose? So glucose is now concluded to have 5 alcoholic groups and 1 aldehydic groups. All of them are now present at different carbon atoms. How do we actually know that? Because there are various stereoisomers possible for that. And we know if there is only one stereoisomer present, if there is one chiral center present, that would give us 2 stereoisomers. And how many chiral centers are present here? So carbon center 1, second, third, fourth. 4 are present. So 2 raised to the power n, where n is number of chiral centers. That's 2 raised to the power 4, which is equals to 16. So there are total 16 possible configurations are possible. Only one of them is known as D-glucose. I'm sure you must have tasted or had D-glucose bought from the market also, which is commercially known as D-glucon. है ना? Glucon बोलते हैं. So that D actually represent its configuration. Capital D, make sure you are not writing it as small d. Capital D, small d, they are different. Capital L and small l, they are different. So let's move ahead, everybody, and then discuss what is this configuration. There are 2 types of glucose known to us. One is called alpha isomer, the other one is called beta isomer. When you take glucose, when you take a glucose, and its melting point noted is approximately 419 Kelvin. Make sure it is Kelvin. It's not degree Celsius, it's Kelvin. How do we prepare it? You have to crystallize the solution of concentrated solution of glucose at 303 Kelvin, which is 30 degrees at room temperature. Take a solution of glucose. Keep it for crystallization. What you obtain will be alpha glucose. But if you want to obtain beta glucose, I'll tell you what is alpha, what is beta here. Its melting point is not very different, just 4 Kelvin ka difference, 423. They can also ask you, examiners can also ask you which one has higher melting point. Beta glucose has higher melting point, although the difference is not much. To crystallize this, we need to heat and saturate glucose ka solution and then crystallize at 371, which is approximately 98 degrees Celsius. At that temperature, the structure of glucose will be beta glucose. The isomer obtained will be beta glucose. You want to see the structures of alpha beta glucose? Now we can now see that there are 2 types of isomers possible. Why am I saying isomers? Because the molecular formula is exactly the same. Make sure that both of them are D and D. This is also D, this is also D. The only difference is alpha and beta. So that was the structure we are known so far about the glucose. When you draw its Haworth projection or cyclic structure of glucose, this is the alcohol unit which attacks on the aldehyde unit. Let me write and show it to you. Let me pull in a slide and then explain. So suppose you have CH3 C double bond oxygen hydrogen, and I attack it with ROH. This is an aldehyde. I am attacking it on alcohol is attacking on the aldehyde unit. So since carbon cannot form for more than 4 bonds, the pi bond has to open up. Oxygen can take care of the negative charge very well. It becomes a negative charge species. This is R with oxygen and carrying a positive charge. Okay, oxygen, whenever it forms 3 bond, it becomes a positively charged species. In the same molecule, there is a positive and negative charge present. If they can help each other for neutralizing each other, then that reaction is going to occur. That reaction is going to occur. This is a basic site. Oxygen minus is a basic site. That's a base. And this is an acidic site. This is an acid. Intramolecular acid base reactions are so quick, so fast that immediately this oxygen minus is going to take up this hydrogen, make this oxygen positive also neutral, oxygen minus also neutral. So what will be the product obtained? The product obtained will have one OH connected and one OR connected to it. This is known as hemiacetal. Hemiacetal. So you started with an aldehyde, attacked it by an alcohol. The product obtained has one OR unit, one alkoxy unit present on it. This is known as hemiacetal. Hemiacetal. So if you repeat this exercise, then you obtain a complete carbon CH3 with OR on both the sides, OR and OR. That becomes acetal. This is a hemiacetal at this situation. That otherwise, if I repeat this process of attack by alcohol, you will get acetal. You will get an acetal. Now suppose this alcohol and aldehyde, both the units are present in the same molecule, then also the intramolecular attack is going to happen. What kind of attack it is? Intermolecular, because these are two different molecules attacking on each other. Correct? Now if I have a molecule which contains C double bond O aldehyde unit, and there are so many carbon here and there, so many carbon atoms are present. Suppose there are so many carbon atoms are present and there is OH present. Sir, it looks like the same reaction can occur here too. This oxygen pair is going to attack over here and aldehyde is lost. Aldehyde group is vanished, is distinct, is gone, it's over, it's no more present. Sir, it looks like I will have CO minus hydrogen present and then there is some sort of ring formation. Do you notice that some similar kind of reaction is happening here? What I just did here, similar reaction is happening, but the result is a cyclic structure. The result is a cyclic structure. Again, acid-base reaction. This is going to pick up this hydrogen. This becomes a neutral, and then the structure has got OH with the hydrogen, cyclic structure with oxygen connected here. Sir, it looks exactly like acetal. It looks exactly like hemiacetal, but that's a cyclic structure. This becomes cyclic hemiacetal. Cyclic. This is cyclic hemiacetal. This is cyclic hemiacetal. This is the same chemistry occurs in glucose also. The CHO group is no more present. CHO group is not free. CHO group is attacked by its own member of OH group, on its own member, which is a part of its group, which is a part of its structure, attacks on the CHO group is gone. It is converted to cyclic structure now. Now let's go back and see how it has happened. I'm sure now you are able to understand this oxygen attacks on this carbon and then form the cyclic structure. During this attack, as a result of this attack, the C double bond OH, which was a planar molecule initially, it was a planar site because C double bond OH or C double bond OH. I want you to tell me what is the hybridization of this carbon center in the comment box. Hybridization is sp2. Right, everybody? sp2 means planar geometry. If attack happens, it can happen from the top or it can happen from the back. As a result of which, there are two isomers, there are two stereoisomers going to be generated. One is alpha, the other one is beta. So it means that the configuration here, there is not going to change. They are not going to change. They will remain the same. Exactly, they remain the same. So if I say this is my carbon number 1, carbon number 2, carbon 3, carbon 4, carbon 5, and carbon number 6. These are the chiral centers, carbon 2, 3, 4, and 5. They are the chiral centers, stereo centers. So they will have their position of H and OH exactly the same. They are not going to be disturbed. But this end molecule here, pardon guys, this molecule here, the secondary one is going to attack, not the last one. This alcohol is anyways going to attack here. This is going to attack. So why not the last one? This will actually form 7-membered ring, which is not very stable. That was also possible. That was also possible. But when this oxygen attacks on this carbon center, there will be 6-membered ring generated. But if this oxygen is going to attack, it will result into 7-membered ring. And we know 7-membered rings are less stable than 6-membered ring due to ring strain. So oxygen from this site attacks here. What is it going to do, guys? It will be generating. It will be eventually generating alcohol and acetal, one of the acetal. So if I number this as carbon number 1, this is my carbon number 2, this is my carbon number 3, carbon number 4, carbon number 5, carbon number 6. This is carbon number 6. So this CH2OH remains as it is. The 5th carbon hydrogen is on the left hand side. Oxygen has already attacked, so OH is disappeared. OH is gone. Moving to the next one, this configuration is still the same. Moving to the third one, this is again the same. Moving to the second one, this is again the same. And now the first one, which was originally planar, now has got two type of configuration. One side you may have OH on the right hand side, and in the another stereoisomer you may have OH on the left hand side. Now listen to me very carefully. Listen to me very carefully. If you have OH on the right hand side of the cycle, this is known as alpha isomer. And if you have OH on the left hand side of the ring, this becomes a beta isomer. These two are known as anomers of each other. If the configuration on the first carbon changes, if the configuration of carbon number 1 changes, is known as anomeric structure. They are anomeric. Anomeric structure. They are anomeric structures to each other. They are two anomers of each other. Remember, alpha and beta glucose are anomers of each other. Understood, guys? Okay. Now, this is the cyclic Haworth projection of glucose molecule. How we actually draw the structure. Whatever is on the right hand side, is drawn above, and whatever is there on the left hand side is written below. So what is there in the alpha? We have OH on the right hand side. So let's refer to the structure. We have got alpha and beta structure. See, I said OH molecule was on the right hand side, it will be drawn above, it will be drawn below. This was on the right hand side of the cyclic structure. Hydrogen was on the left hand side of the structure. Likewise, this OH is on the right hand side. This is the hydrogen, OH and CH2OH. CH2OH will be always shown above. So OH on the right hand side, OH on the right hand side, H on the right hand side. Let's see that confirms the structure or not. OH on the right hand side, OH on the right hand side, hydrogen on the right hand side, again OH on the right hand side. Make sure you draw CH2OH on the top of the 6th carbon. We have to start drawing the structure in the form of pyranose. You are known to the fact that if there is no oxygen here, if they all were carbon atom, this is known as cyclohexane. If I just remove one of the carbon atom here and replace it by oxygen here, this becomes a pyranose. Hence these structures, these glucose projections are also known as a pyranose. This becomes a pyrene ring. And since it is a glucose gas structure, these are aldose. These are given a name pyranose. When you decorate this ring, pyrene ring, by loads of OH, hydrogen and CH2OH, this is being decorated by HOH, HOH, OH, H, HOH, OH and CH2OH. Therefore, this is known as pyranose. What kind of structure it is? Glucose. So the overall name of this molecule will be glucopyranose. The name will be glucopyranose. And we know since OH is on the bottom and right hand side of the cyclic structure, this is alpha. But make sure it is D. It is D isomer. Alpha anomer of D isomer glucopyranose. Understood, everybody? Similarly, you can go to this side, which is fructose. Fructose ka structure is 5-membered ring, including the oxygen atom. So this is my carbon number 1. This has OH on the right hand side. If OH is on the bottom, right hand side of the ring, if it is on the bottom, this becomes the alpha structure, and exactly opposite. So if OH is on the top, this becomes a beta fructose. Right, everybody? And this ring is known as a furan ring. This ring, I am sure you must have read this ring, this molecule in aromatic chemistry also, which is known as furan ring, which is known as furan ring. But when you remove these or saturate this ring, this becomes tetrahydrofuran, tetrahydrofuran. And when I decorate this molecule, instead of hydrogen, there are CH2OH, oxygen H, OH, H, OH, H are present. This becomes fructofuranose. This becomes fructofuranose. Remember, if the configuration at carbon number 1, the very first chiral center is different, rest everything is the same, alpha beta are obtained, alpha beta anomers are obtained. Like in the case of glucose, alpha beta are available. Similarly, we have fructose also at alpha beta anomers. So they may ask you in this question, what are anomers? The configuration changed at the first chiral center of glucose or fructose. This definition is not just limited to glucose and fructose. Any kind of chiral compound, if they are different, if the two chiral compounds are different at the configuration, in the configuration of the chiral center number 1, they are known as anomers of each other. Okay, guys. Now, we know very well that when sodium amalgam in ethanol is added, what is it going to do? It will perform a reduction reaction. So what is going to happen to aldehyde? Aldehyde will undergo reduction to form CH2OH. This is glucose. This is known as glucitol or sorbitol. I suggest you to remember these names, sorbitol or glucitol. Rest everything is ditto, same. No conversion, no change at all. This we have seen already. When you treat glucose with HI for prolonged time, prolonged heating, everything is converted to alkene. N-hexane is obtained. Done, guys? Understood? These are the few reactions of glucose. Now, we can also study few reactions of carbonyl, signature reaction of carbonyl. Let me help you with the revision of that reaction first of all. Suppose you have an aldehyde and treating it with hydrocyanic acid, HCN. This cyanic part, CN part, attacks on the carbonyl center, will form RCO, hydrogen and oxygen. They are going to eventually form a bond and cyanide is obtained. This is known as cyanohydrine. Cyanohydrine. Again, this attack of Cn minus is attacking is happening on the planar carbon, C double bond O carbonyl carbon. There is a chance to get two type of isomers again. There is a chance to get two type of isomers again. So now again I can have OH on the right hand side or OH on the left hand side. That's a signature reaction of carbonyl compounds. You must have studied in aldehyde, ketone and carboxylic chapter. Okay. So now if I do a Stephen's reduction, if you do not know what is Stephen's reduction, let me help you with that. Starting with cyano, RC triple bond and reacting it with SnCl2 in the presence of HCl and then finally adding water. What happens? This gets turned to an aldehyde. This turns to an aldehyde. This is an aldehyde obtained. So when I do this reaction, this is known as Stephen's reduction. If I treat the compounds obtained with SnCl2 in the presence of HCl, you will obtain heptagonal or aldohexose. This is aldohexose. You will obtain cyanohydrine compound. Now if Stephen reduction is done, we will get heptagonal. Heptagonal. Alright everybody. Next. So this is the structure of cyclic structure of glucose. We can treat an alcohol with any alkylating agent, so to make it ether. So there are various alcohol units present. If I treat it with one ethanol, there is another ethanol unit is present. In the presence of H plus, if I am going to attack, what is the product going to be occurring, guys? This OH will get protonated. Remember the carbon number 1. This will be protonated and ethoxyca oxygen is going to attack. That will replace OH by ethoxy. That will replace by OET. This will be producing a byproduct. That's water. That's plain reaction. As I said, you can also treat it with alkyl halide by just making it an alkylated product or an ether. This is also ether. Sir, how is it an ether? Sir, this is a carbon which is connected with oxygen and then ET. What is ET? CH2, CH3. So carbon, oxygen, carbon, this framework is ether, right? So this is an ether. Here also you can produce an ether, but make sure if you react it with CH3I, methyl iodide taken in excess, all these alcohol units are going to transform into ether, 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 ether. But in this reaction, first reaction, treating it with ethanol in little bit of acid, only carbon number 1 ka OH will be converted to ether, ether analog. Rest alcohols are untouched. Rest of the alcohols are untouched. Okay, now acylation can also be done. So to do acylation, we have just looked at this reaction ROH upon treatment with CH3 C double bond O, oxygen C double bond O, CH3. This is acetic anhydride. This alcohol will attack over here and you will obtain acylated, है ना, pentaester is obtained. Acylated, all these alcohols are changed to OAC, 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 OAC and OAC. All these alcohols are converted to their acetate, their pentaacetate. So byproduct obtained is here 3 moles of acetic acid, another mole of acetic acid, and acetic acid is obtained. How? Because there are various, how many? 1, 2, 3, 4, 5, total 5 of these alcohol units are present which will be attacking on this. So as a result of this OH attacks here, this group is going to leave. This will be one of the acid ka product. One part of this molecule is attached to alcohol, rest of the part will become an acid, which is written over here. All right, everybody. Next. Now glucose discussion is done. We have discussed how the glucose ka structure was elucidated. How do we know it's a straight chain molecule? How do we know it's a cyclic structure also? How do we know the alpha beta structure? How to prepare them? How we commercially obtain glucose molecule from cane sugar, from sucrose and various other sources? What happens when you treat it with Br2 water? What happens when you treat it with concentrated HNO3? What happens when you treat it with HI with prolonged period of time and heating? All those reactions we have just now discussed. Hydrocyne production, cyanohydrin production also, pentaester production also. Sucrose is a disaccharide, right? Sucrose is a disaccharide. This is one of the starting material to obtain glucose. So sucrose is made up of 2 materials. One is glucose, another is fructose. But how are they linked? That is what is of importance. You can see that this is the fructose ka structure, this is glucose ka structure. This linkage, which looks like carbon, oxygen, carbon, it is an ether linkage, but in this chapter, it's given a special name, glycosidic linkage. So what is this position over here? We have to start the numbering, number 1, 2, 3, 4. So we are utilizing the, or if we start the numbering from this end, since it is beta, so carbon number 1, this is carbon number 2, 3, 4, 5. These are the 5 and plus 6th carbon here. So C2 of fructose is used. C2 of fructose is going to be used, the glycosidic linkage. This has to be beta D fructose C2 linkage. And for C1 linkage, this is carbon number 1 of glucose, carbon number 2, 3, 4, 5 and 6th carbon. This is carbon number 1 of glucose unit. Which glucose it is? Alpha D glucose. What is this unit, guys? Since you are seeing that originally OH, when it is on the bottom, OH is on the bottom, but it has to be on the right hand side. That makes it alpha fructose. Since it is twisted, I'm starting the numbering from this end. This is carbon number 1, carbon number 2. This is beta D fructose. Be careful, it's beta D fructose. So C1 of alpha D and C2 of beta D fructose. These are the 2 molecules connected to each other via glycosidic linkage. Do you notice that on the first position, this is the first position of glucose or fructose, this is the first position of, this is the first position, this is the second position of fructose, this is the first position of glucose. At none of the positions, OH is free. This OH, both of them are now, they have produced an anhydride, they have removed water, they have condensed into each other. Therefore, sucrose is known as non-reducing sugar. This is very important question, guys. Why is sucrose a non-reducing sugar? Because there is no free OH present. So there are some more OH present. No. If this carbon number 1 and this carbon number 2 have no free OH present, then this becomes a non-reducing sugar. Non-reducing sugar. Sucrose, what is this property? It's a dextrorotatory. Take it in water and then you see where in which direction it rotates. A plane polarized light, it rotates in clockwise direction, hence it is given a name dextrorotatory. But interesting point is when you hydrolyze sucrose, with the help of some amount of acid, it will dissociate, it will hydrolyze into glucose and fructose. Glucose rotate in clockwise direction, fructose rotate in anti-clockwise direction, and as a result of the two, as a result of the competition, overall mixture, hydrolyzed mixture, rotates light in anti-clockwise direction, in an invert direction, in the reverse direction. Hence, sucrose is also known as invert sugar. Very important question. Why sucrose is known as invert sugar? Because its mixture gives you inverse rotation than when it is in intact form. So hydrolysis of sucrose brings a change in the sign of rotation from dextro to levo, and the products named as invert sugar. Very important question, guys. Done. Let's proceed. Now lactose. Lactose, it looks like it is made up of two type of sugar molecules. This is beta D galactose. Be careful, it is beta D galactose, not glucose. The first carbon which has OH, which had OH on the top, now it is condensed, and this side we had beta D glucose which has beta OH on the top. So this is my carbon number 1, 2, 3, and fourth carbon is linked to first carbon of galactose. So what is this? Beta D glucose, beta D galactose. They are now condensed with each other. Water removal happens and glycosidic linkage is obtained between carbon number 1 of galactose and carbon number 4 of glucose, and both of them are beta D, galactose and glucose. This is more commonly known as milk sugar. Lactose is present in milk. Some people have lactose intolerance because they cannot hydrolyze lactose. They lack one of the enzyme which is used to hydrolyze inside human body. It is composed of beta D galactose and beta D glucose. The free aldehyde group may be produced at C1. So do you notice that here there is C1 present, here also there is C1 present. Here is there any OH free? No. Is there OH free over here? Yes. If OH at carbon number 1 is free in glucose, then it will be a reducing sugar because it can be oxidized and it will reduce others. This can act as an oxidizing agent. This can undergo an oxidation to produce an aldehyde. Therefore, it will be helpful in reducing others and oxidizing itself. Such kind of reagent which will oxidize themselves and reducing others, they are known as reducing agent. Hence the name reducing sugar. So lactose is a reducing sugar. But is sucrose a reducing sugar? No. All right, everybody. Okay. Now comes to the maltose. Maltose is also a disaccharide. It's also a disaccharide which is made up of two glucose units. There are two glucose units. The first one is the C1 carbon of glucose. This is the C1 carbon of glucose. Which kind of glucose it is? Since OH is on the bottom, it is alpha. C1 of alpha D glucose. And C4 of another alpha D. This is carbon number 1, this is carbon number 2, carbon number 3, carbon number 4. So 1 and 4, the connection is in between carbon number 1 and carbon number 4. Both of them are beta glucose. So what's the product of maltose hydrolysis? It is glucose, beta D glucose. So this OH is free at position number 1. Please check, this OH is free at position number 1. Therefore, it is a reducing sugar once again. It's a reducing sugar. So we have seen two type of reducing sugars. One is lactose, the other one is maltose. Okay, very good. You should be in a situation to write down their structures to justify why they are reducing sugar and why they are not. Okay. Now we have polysaccharides to discuss. The very first one is starch, which has got two components in it. The one is called amylose, the other one is amylopectin. This is a very large molecule, complex molecule. This is relatively simpler. So it contains a large number of monosaccharide units joined together by glycosidic linkage. That's it. That is what we are studying in this entire chapter. So you please notice that this is my carbon number 1 of glucose of alpha D connected to another alpha D, but the fourth carbon. So carbon number 1, carbon number 4. This is known as 1,4-alpha glycosidic linkage. 1,4-alpha glycosidic linkage, which is actually happening in amylose. So far we are not discussing amylopectin, so don't be confused. This is just the structure of amylose, where you have linkage between carbon number 1 and carbon number 4 of glucose of two different glucose units. This gets continues. This gets continues. Hence, it's a polymer structure. It's a polysaccharide. It's a polysaccharide. What kind of linkage it is, guys? Once again, C1, C4 glycosidic linkage alpha. This is known as alpha, and we need one kind of enzyme which breaks this molecule, alpha amylase, which breaks this molecule into glucose unit, and hence we receive energy. Okay, but amylopectin is relatively complex. As I said earlier, there are not just 1,4 linkage, there is 1,6 linkage also. There is 1,4 linkage, on top of it 1,6 linkage is also present, and that 1,6 linkage makes it complex. That 1,6 linkage makes it complex. So alpha D glucose, in which the chain is formed, in which the chain is actually formed. Let me pull this slide a little bit up so that you can read it. Now, alpha D glucose units in which chain is formed by C1 C4 glycosidic linkage. Wherever branching, where is the branching occurring, guys? Branching is occurring at C1 by C1 C6 ka linkage. C1 C6 linkage, and that becomes a challenge for a human body to digest, to break this. We lack one particular enzyme, and we are unable to digest that. So that becomes a challenge for us. All right, everybody. What is glycogen? This is again a very famous question. In what form glucose stores in animal body? It is glycogen. Glycogen, carbohydrate stored inside human or animal body. You should know that. Okay. And it's also known as animal starch because the structure is similar to amylopectin. As I said, amylopectin and cellulose, they have similarity. Humans cannot digest cellulose because cellulose has got complex structure, and we lack that enzyme to hydrolyze cellulose. Okay, again it's present in liver. What is the location of glycogen in human body? It's liver, muscles and brain. When the body needs glucose, enzymes break the glycogen into glucose. Glucose is not directly present inside. It's actually gone inside. We take the sources of glucose that breaks down to produce glucose, and then glucose gets collected, accumulated in the form of glycogen. And whenever you need, glycogen breaks to produce glucose. This is the structure which looks similar to amylopectin. There is 1,4 linkage as well as 1,6 linkage, complex molecule. Alright. The cellulose, as I just discussed, it occurs exclusively in plants and most abundant organic substances in plant kingdom. Okay. And it is predominant constituent of cell walls. You know, plant walls are plant cells are having cell walls. Cell walls are generally having cellulose coated on it. It's a straight chain, of a straight chain. How is this molecule made, guys? I just have discussed. This composed of only beta D glucose. What kind of glucose? They can ask you this question. What kind of glucose is here in cellulose? It's beta D glucose. And the connection is in between carbon number 1 and carbon number 4 of glucose. And it continues and it becomes a staircase like structure. Staircase, because carbon number 1, carbon 4, again carbon number 1, carbon 4, again carbon number 1, carbon 4. So it becomes a staircase like structure. Very complex molecule. Okay, now let's come to amino acids. As the name is suggesting, the molecule which contains amino group as well as acid group in it are amino acids. So the general structure would look like a molecule having amine, as well as acid. Both the groups are present. Sir, it looks like this is an acid, this is a base, so they should react with each other. Yes, they do react with each other. And the real structure of an amino acid is not this one, rather it is having charged structure, NH3 plus and COO minus. Any molecule having a positive and negative charge together present in the same molecule is known as a Zwitter ion. Zwitter means two, double. It has two charges, one is plus and other is minus. It is opposite charges, they are opposite charges. This is given a name, Zwitter ion. Zwitter ion. Zwitter ion. Not just the amino acid, any molecule having plus and minus together in it is known as Zwitter ion. Okay, guys. If COOH and NH2, both of them are present on the same carbon, they are known as alpha amino acid. They are known as alpha amino acid. This is alpha amino acid, and R group can be alkyl or aryl or any sort of chain, any sort of carbon based chain. So then we are going to discuss what is how they are actually present in the medium. This is what I just told you. They exist in the form of zwitterion in a medium, and they can now behave as acid or base whenever they require. So this can act as a H plus. They can accept H plus, and this side can donate H plus. So amino acid can act as a source of acid as well as a source of base whenever whatever function is required. Amino acids does for us. Amino acid does for us. Amino acid has got a class. One is called neutral amino acid, the other one is basic amino acid, and the last one is acidic amino acid. How do we identify? Based on the total weightage of acidic or basic group. You know, amino group is basic and COOH is acidic group. If the number of acidic and basic groups are equal, that's a neutral amino acid. If the number of acidic groups are more than the basic, overall it's acidic in nature. And if the number of amino groups are more than acidic groups, then it becomes a basic amino acid. So I have just told you, if the number of acidic site, what are those? COOH. If they are more than NH2, then it will be acting as an acidic amino acid. To name a few, glutamic acid and aspartic acid. And if it is neutral, X and Y, they are same. What is X? The number of carboxylic, number of COOH, number of NH2. NH2 represent Y group and this group represent X. If X and Y, they are same, then it's a neutral amino acid. Basic will be the one which has got more amino groups. Example, histidine, lysine and arginine. Neutral ones are like valine, isoleucine, leucine. These are the examples of neutral amino acids. Now what are proteins? Proteins are made up of amino acids, and amino acid together will form a peptide chain. Peptide chain will form polypeptide chain. Polypeptide chain may form proteins, and they can also form enzymes. So proteins are the polymer of alpha amino acids, and they are connected to each other via peptide bonds. Like we had glycosidic linkage, which is originally ether linkage, and in the case of amino acid getting connected to each other, there is amide linkage, which is given a special name, peptide linkage. You can see over here, these are the two amino acids present. I can condense these two to make one polypeptide. So now this nitrogen, which was originally having NH2, now it has got connected to another carbon. Now please notice this has got NH2 free, this end has got carboxylic acid free, but what is this function group? What is this organic molecule? What is this function group? This is known as amide, but amide in the case of protein, they are known as a peptide. When two amino acids come together, they will generate one amide linkage, that is known as peptide linkage. This is a peptide linkage. Okay. So two amino acids, when they come together, they generate a dipeptide. Although the number of peptide bond is just one, overall the molecule is generalized as dipeptide. If three amino acid comes and then condense with each other, removes water, that will generate a tripeptide. So the name is simple. Di represent 2, tri represent 3, tetra represent 4, and if many amino acids come together and they undergo dehydration to just form amide linkages, that will be polypeptide. And similarly, when you hydrolyze polysaccharide, you generate monosaccharides, various monosaccharides, and when you hydrolyze polypeptide, you will be obtaining various amino acids, which is the starting material for the peptides. Now this is what I said. Glycine has got NH2 and CH2. Glycine is one of the amino acid which is not chiral. Only one of the amino acid which is not chiral. Now I will be taking out this molecule, this H2O. When it comes out, this becomes my amide linkage, which is given a name, peptide linkage. So what is this peptide, guys? Dipeptide. And how do we actually represent the sequence of protein, sequence of amino acid in the protein? The first one has got a 3 letter code. है ना? Amino acid have got one single letter and 3 letter code. So glycine is represented by GLY and alanine is represented by ALA. So this is GLY ALA. By reading just this name, you should be able to identify that GLA is present in the left and ALA is present on the right hand side. Similarly, let's do this exercise. Alanine gets condensed with glycine and it forms alagly. Alagly. This side is alanine, the other side is glycine. Understood, everybody? This is a dipeptide. This is also a dipeptide. You can continue doing this dehydration, removal of water, condensation, and form a polypeptide. Okay. So let's do some classification of proteins. There are 2 types of proteins. One is called fibrous, the other one is called globular protein. The fibrous proteins, they run parallel. They run parallel and held together by hydrogen bonding. So one of the, it's just like cotton candy. It's just like cotton candy. So they are fibrous in nature. So polypeptide, they will run together. One polypeptide chain is like that. The other polypeptide chain is also moving around, and they are connected to each other with the help of some sort of van der Waals forces or hydrogen bonding. Okay. So this is insoluble in water. Remember, it is insoluble in water. Examples: keratin, which is present in your hair, in your nails also, and myosin. These are the 2 examples. They may ask you in this examination that what are the 2 examples of the fibrous proteins. Okay, and what are their properties? They are not soluble in water. On the other hand, globular proteins, they are chain of polypeptides. They are coiled together, just like a spring. They are coiled together, one over the another, just like braille, choti, hai na? They are brailed to each other. They are coiled around each other to give a spherical shape. And it is soluble in water. For example, insulin and albumin. You know insulin. Some people take insulin ka shots, hai na? Okay, those are water soluble. Now you can remember insulin shots are taken, and that gives that goes around goes via blood and get delivered to the desired position. Okay. Now protein has got various kind of structure. One is called primary, secondary, tertiary, and quaternary structure. How does primary structure look like? When you have amino acid connected to each other in a straight chain, that represent a primary structure. That is just the primary structure when amino acids are connected in a straight chain. But secondary structure, or one more thing, the sequence of amino acid is said to be the primary structure. Sequence. You have 1, 2, 3, ella, gly, well, all those when they are connected one after the another, that becomes a primary structure. Okay. There are 2 types of structures again. One is called alpha helix. Do you see a spring-like structure? And then there is a beta sheet-like structure. Beta sheet. In the same framework, they have got some sort of hybridization. See this alpha. This alpha helix also has got some sort of hydrogen bonding to stabilize them. This is the reason why people have curly hair. Some people have curly hair because these are proteins in the hair. They are joined together, they are connected together via hydrogen bond, or sometimes disulfide bonds also. Okay. So there is secondary structure, as I just showed you, alpha helix and beta pleated. Beta pleated is just like sari. You know sari? Sari, you make a pleat like that, or just take an example of curtain. One curtain has got one plate over the another, so that is the similar kind of structure of a protein. Beta pleated structure. Alpha helix structure is just like a spring, so they are held together via hydrogen bond or disulfide linkages. So secondary structure arises due to the folding of backbone of polypeptide chain by hydrogen bond, and alpha helix is a right-handed screw structure. Screw structure. Remember, it coils like that. Right-handed screw structure. And in beta pleated structure, peptide chains are stretched out to the maximum extension and then joined to the via intramolecular hydrogen. Within the same, I'll show you the structure once again. Here again, beta sheet, they have got intramolecular hydrogen bonding. This is the structure. So they fold to the maximum. They go via maximum extension, then they start folding. So they are parallel, one after the another. This is a parallel one. These two are parallel. They are said to be parallel, but this one will be set to anti-parallel because it is moving in this direction and the next line moves in this direction. So obviously, yes, these are their properties. This line starts from here. The polypeptide chain starts from here. It goes in this direction and then it has to go in the other direction. So they two will be known as anti-parallel to each other. The first one and the third one will be known as, the first one and the third one will be known as parallel. Okay. All right. Now tertiary structure is representing overall folding, So you may have alpha helical as well as beta plated. A combination of the two will represent tertiary structure. Combination of the two will represent tertiary structure. Then this gives rise to two type of molecular shapes which we have just learned, globular and fibrous proteins. So tertiary structures are responsible for the generation of fibrous and globular proteins. Okay, so what are the reasons behind tertiary structure? When we obtain tertiary structure, disulfide linkages, van der Waals forces, and sometimes electrostatic force of attraction also can bring the two together and cause it even further folding of the molecule. You can see over here, this is the polypeptide chain which is folding over here, and then they have got polypeptide chain connected to each other via some sort of linkages also, some sort of bonds also, and those bonds can be disulfide linkage. We know cysteine is one of the amino acid which is sulfur containing. There another, if in the chain there is another sulfur containing amino acid is present, so they two will come close to each other and form disulfide linkage and cause better folding. That's the reason people have curly hair. Okay, quaternary structure is a little bit complex, which says proteins are composed of generally proteins which are heavy molecules, and for their molecular mass prediction also, molecular mass representation also, we have a different unit called Dalton. Proteins are composed of two or more polypeptide chains, and each polypeptide chain is known as subunit. For example, hemoglobin has got 4 subunits in it. Okay, all those units are complex. So the spatial arrangement of these subunits with respect to each other will give it a structure, quaternary structure. Hemoglobin has got 4 units, 1, 2, 3, 4, and all of them they're arranged in such a fashion that they provide us a quaternary structure to the hemoglobin. Okay. Now, what are nucleic acids? Very important part of this chapter. Nucleic acids are responsible for our genetics. And DNA, RNA, they are responsible. So the nucleotides are the combination of 3 things: base, sugar, and phosphate. Nucleoside are the combination of just the 2 things. So we have got nucleotides which are joined through 3' and 5' phosphate linkage to give polynucleotides. I'll show you 3' and 5' position. There are 2 types of nucleic acid. One is a ribonucleic acid. The other one is deoxyribonucleic acid. On what position there is deoxy? On 2' position. When there is no more OH present on second prime carbon, this becomes DNA. Right, everybody? So complete hydrolysis of RNA, what is it going to yield? 3 things. One is pentose sugar, as I said, phosphate, phosphoric acid, and the nitrogen containing base. So base comes first, then sugar, then phosphate. This is the sequence for of a nucleotide. First it has to be the base, then it has to be the sugar molecule, then it has to be the phosphate unit. I'll show you the structure now. So this is a nucleoside which is made up of 2 units. One is called sugar and the base. As I told you, base always comes the first. Then comes the sugar. This is my overall base. This is the structure of the base. This is the base connected over here. Base can be of 2 types. Now we can have 2 type of purine and pyrimidine. These kind of 2 bases, and this is the sugar ka structure. This is my sugar can be now again various sugars that we have learnt. This is my carbon number 1, carbon 2, 3, 4, and 5. Do you see on carbon number 2 you have OH? Yes. So this will be a backbone. This will be the starting material for RNA. But when we move to this side, nucleotide, BSP, base, sugar, and phosphate. This is the base here. We move from left to right. Left to right, base, then sugar moiety, and then comes the phosphate. Phosphate is making it in a phosphate ester. This is CH2OP, this is phosphate ester. Where is it connected? On the 5th carbon. Where is it connected? The base is present on the 1st carbon. This much you should know. You should be in a situation to draw these. So sugar can be ribose or deoxyribose. This is ribose sugar and this is deoxyribose sugar. Ribose, just like glucose, this is called ribose sugar. Okay. Now, the nucleic acid may contain four type of phosphate esters. The one is, this is the real structure. This is the structure of phosphoric acid. If it gets connected to the sugar moiety, it will generate phosphomonoester if there is only one R present. There are two more OH groups present. Let me go to the slide back again. There is only one. This entire group is considered as R. So what is this, guys? This monoester. I can take the same unit once again. This will become diester. I can take another unit here. That will become triester. Make sense, everybody? So you can have phosphotriester also. Diester, monoester. This is the structure of phosphoric acid. My question to you, you have to answer in the chat box. What is its oxidation state and what is its hybridization? What is its hybridization, oxidation state? Yeah, that's all. Okay. All right. Yeah, there are two type of nitrogenous bases. I told you sugar is only one kind, ribose or deoxyribose. The nitrogenous bases are of two type. One is called purine. Purine has got two rings. The pyrimidine has got only one ring. Let me pull the slide a little bit up so that you can see the structure. This has got two rings. These are called heterocyclic rings. Hetero means when you have any atom apart from carbon a part of the ring. Apart from carbon as a part of the ring becomes a heterocyclic compound. This is a pyrimidine. Now we are going to derivatize it. Derivatize and obtain various purine and pyrimidines. Okay. So let's see what are those. So there are a variety of nitrogenous bases which are found inside human body. One is called adenine. The other one is guanine. Then cytosine, thymine, uracil. You should be remembering them by their acronyms also. A, T, G, C, U. A actually likes T. G actually likes C. So A, T and G, C, these are the two combinations we figure out in DNA. These are the two type of combination we find in DNA and in RNA. So adenine and guanine, these are the derivative of purine because they have got two rings in it. These are purine bases, and 1, 2, 3, these are pyrimidine bases. They may ask you this question to draw their structures also. You should be having those on your tips. Okay. That's all from Biomolecules chapter, everybody. I hope you have got all the information required for your exams and this video you have found useful. I will bring to you another video for your summary series for your next chapter. Till then, guys, stay safe and keep studying. Bye, guys.